Good afternoon, everybody. Just a little bit of an overview of what I'd like to talk about today. This is really meant as an introduction on the power of a data analytics platform combined with product characterization, new therapy process development data, and the massive analytical data that we actually generate as we're working on developing these processes. A quick introduction to where I live in the Sartorius organization and our accelerated cell line development. Then looking at these platform processing capabilities that we have for design of experiments and analytics. And then I give you a view of a couple of quick case studies at a high level topic. For Sartorius has 8,000 employees in 30 different countries and $1.6 almost billion in revenue. We're made up of two primary divisions. One is our research and tools division, which is the lab products, and our Bioprocess solutions division, which I'm a part of, and in which our cell line development technologies are housed. This is kind of a view of the world that we live in, which is cell line development, cell banking, and GMP manufacturing of cell banks. And we provide everything from a transfected cell all the way along to a GMP cell bank. And in fact, we are very involved, and we gather huge amounts of data along the way here. And of course, we've got tools and technologies to kind of fit against all of these cell lines that we are developing and banking. The piece I'd like to focus on is how we apply a data analytics platform to the process data, to the conditions of culture, and then to the analytical and characterization data that we're gathering as we go through the production process. And really, I think some of the conversations that we had earlier from the two previous presenters underline the power of being able to take all of these individual data points and process them parallelly, and then understand what the relationships between them are. Because very often, for example, we find that you don't have picking the most productive clone or the best grower is not actually the best solution when you get to a product which you're selecting for the clinic. So we look at our platform as having three major capabilities. The first part is designing an experimental model which will optimize the data that we're going to get back out of it. Then using that data in a reverse platform to model, and then deploying the predictive nature of that to get us to real-time process monitoring, in-process changes, and optimization of that. And there's been quite an explosion of data, as you guys all know, from the advent of process sensors, spectrometry and chromatography data that's coming off the characterization of that process and product. All of the lab data that's been generated around in-process controls, measurements, and release data, and then raw material properties, and lastly, the quality data. And so the question really is, what kind of insights are we going to get from processing all of this characteristic data together? So we can operate at very different scales. In the research environment and the development environment at the milliliter scale, all the way to commercial manufacturing and gathering process data at the hundreds to thousands of liters of scale. So we were sitting around thinking, if we just knew as much about the things that drive a process as we do about the variables that drive something that's part of everybody's life, like the Dow Jones, how much better off would we be at manufacturing? How much better off would we be at development? And how much better off would we be at getting the medications to the clinic and to the patients faster. And here we are today with a curve that is similar to the one I just showed you, but this has a whole bunch of known data points, but up to now very little known about what combination of these drive critical quality attributes of a product. And so if you were to have access to a platform like the Sartorius Simca platform, what kind of insight would you get from that processing power, and then what decision would you make about which candidate to move forward? And there's a strong business impact as well. These platforms can process your process data, cost of goods data, product quality and similarity data, and all of these things together will give you the insight to develop the product at the stage it's at for the right cost structure and of course quality. So just to give you a couple of views into applications of the processing platform. This is a typical project for us where we would develop a cell line all the way to the research stage in a 4-month window, and then get a master cell bank transferred back to a contract manufacturer within 10 months. And this happens routinely for us, but only with the ability to do sophisticated data processing, data analytics. And every step or every day we save, or every candidate that we don't move forward as a clone because of the data analytics that we perform on it, allows us to meet those timelines routinely for customers. So I'm just looking at and this is a design of experiments that we did on for a biosimilar development product in an ambra platform, where we did a design of experiments for screening, followed by a multi-week perfusion process where we looked at things like the cell growth profile, feeding strategies, and also profiling of glycan profiling and charge variants, etc. All of the resulting data was then loaded up into our Simca platform with a view to extracting the optimal conditions under which to pick the right clone and drive forward with the right candidate. And so using the platform, we were able to take down charge variants by 44%, reduce the glycans by 17, and then get volumetric productivity up by 11%, all of which raised our ability and the probability of success in the later phases. A second study where we were doing accelerated production of high density cell banks, where we could end up with high concentration cell banks frozen in bags, and then that could be thawed and directly inoculated into the cell culture system. Again, this doesn't happen without the ability to look at all of the process conditions that are considered to be critically driving the process, pick the optimal conditions that get us to this result. And lastly, looking forward, there are some opportunities in the next generation therapy, CAR T manufacturing, for example, where we're looking at the cell expansion. So the cell expansion for this CAR T product is a 3 to 10 day process in an automated bioreactor system with huge variability because of the patient variability and the raw material. And then different process settings are required to produce the optimal result. The platform allowed us to create a map of the critical process conditions that were driving the process so that it can be optimized, and ultimately allowed us to gather information that would help us intervene in the case that we noticed a detection issue. Similarly for lentiviral production, higher volumes of course, but we had a 3 to 4 day automated bioreactor process, and again, with critical process parameters defined, we could look at how those process parameters were driving the production and allow us to forecast how these processes deviated and intervene before we had a disqualifying result, so early fault detection. And so, in principle, the processing that we do allows us to not just create optimized conditions, but also predict where the product is going to come out, and lastly, make changes in real time that will allow us to better the result that we're looking for from a given process. Lastly, this is just an example of kind of the end stage of the analytics platform, where we've developed, an analytical toolbox in the form of an app, which allows you to look at clone selection data and use an analytics program to actually compile, aggregate, and do the analysis on the data, so that you don't have to be you can rule out human subjectivity in the choosing process. So for us, the takeaway message is we have everybody's working on some form of biological factory in the form of a cell, whether it's a host cell for viral production, or whether it's a CHO cell for antibody or complex protein production. And so the message really is the data that we generate all around these smart factories that are delivering the next generation of therapies will drive smart choices and drive us to get a better result faster and probably cheaper. So in terms of where we're going with analytics, I think we're probably at stage 2.5 here, where we've gathered and stored huge amounts of data, which is retrospective around what happened, whether it's in-process data, whether it's QC data, whether it's historical data across the life of a cell line. It could be chromatography, spectroscopy data. We are very, very good at doing things like investigations, but oftentimes our investigations do have one of two results. Either the conclusions of an investigation are too late to actually do anything about what's happening, and therefore we can only learn from it the next time. Where we need to get to is predictive and beyond. And for that, to be real, we need to have a tool that allows us to aggregate huge amounts of data. So where this is going, and the types of things that we're working on and thinking about right now, are taking all of the quantifiable data that we've got, which is spectroscopy, HPLC, unprocessed NMRs, multiple sets of biosafety and target binding data, and marrying that up with unstructured data, the likes of visual images from things like transmission electron micrographs, high definition images, high definition visuals, and putting it all into one place to understand what's next for the predictability of these products that we're making. Thank you. That's it for me for today.